Today I'm going to talk about bacterial protein secretion and targeting and give an overview of the subject, well, at least cover a variety of different secretion systems. I've borrowed slides from Julia Lodge for some of this, because she gave this lecture last year. I'm getting a bit of an echo there, so turn this volume down. So again, just to remind you where this fits in the course, you're going to have 5 lectures from me on bacterial protein secretion, various systems dedicated to this. This is the first one. Thursday we'll be talking about bacterial flagellum, then later, following week, type 3 secretion, something called ESX secretion, and sortase and LPXCG proteins. Now, before we start, we need to we need to look at what is the issue of what is the issue we're looking at here? What is protein secretion and export and translocation and so forth? Well, if we look at bacterial cell envelopes, they come in 2 major varieties. The gram-positive cell envelope and the gram-negative cell envelope. Sometimes the term monoderm and diderm, these terms are used instead of gram-positive or gram-negative to describe the construction of the cell envelope. So in the gram-positive or the monoderm, there is a single cytoplasmic membrane, no outer membrane. There is this cell wall, which will contain things like the peptidoglycan. And if proteins get across this cytoplasmic membrane, they can get out into the external milieu, they can get out into the cell wall. And effectively in a gram-positive bacterium, secretion, export, translocation, they're all the same thing. You're just jumping across this one membrane. In gram-negatives, we have a rather more difficult situation because we have these 2 membranes, the inner membrane and the outer membrane, spanning the peroplasm, which will in itself contain things like peptidoglycan. And the ways in which these words are used is a little bit variable from one person to another, but some people would say that we should talk about secretion only when we're talking about getting across the whole cell envelope to the outside world. So across the inner membrane, the peroplasm, and the outer membrane in one step. If we're talking about protein export, we're talking about just getting stuff out of the cytoplasm across that inner membrane and into the peroplasm. And sometimes the term translocation, when you're referring to a particular membrane, you talk about translocation across that particular membrane. And so we have translocation across the inner membrane or translocation across the outer membrane. Now, just to give you a bit more of a view of insight into what's in that gram-negative cell envelope, which is going to be the target of this particular lecture. In fact, most of the lectures, 3 out of the 5 lectures, will be looking at gram-negative organisms. We have this inner membrane here with membrane proteins in it. We have the peroplasmic space here with peptidoglycan and peroplasmic proteins in it. Then we have the outer membrane and the lipopolysaccharide on the outside. So the challenge in gram-negative protein secretion is to get stuff all the way across. Now this is quite a busy figure, but it just gives a flavour of the complexity in terms of the variety of different mechanisms, different protein secretion systems that exist in gram-negative bacteria. So this is from a review by Michael Desvaux, who used to work here with Ian Henderson, which he wrote a couple of years back, trying to classify all these different systems and actually come up with unified nomenclature, unified naming systems for them. Now, I'm not expecting you to take much in from just looking at that. The key point is that we can classify these systems into 2 main varieties. There are those where there is translocation across the inner membrane, export across the inner membrane first. The proteins then have a peroplasmic intermediate and then they go across the outer membrane. Or you can have complex protein machineries, protein assemblages, which span the whole cell envelope, spanning the inner membrane, the periplasm, and the outer membrane, all in one apparatus, all in one go, so that proteins going through that route do not have a periplasmic intermediate. They're not exposed to the periplasm. They go through all in one go. So that just puts the same thing in words. Either there is or is not a periplasmic intermediate. That allows you to classify the systems into these two broad classes. So if there's no periplasmic intermediate, and we have this kind of nano machine, and type 1 secretion, type 3 secretion, type 4 secretion, and type 6 secretion all have this property where there is no periplasmic intermediate. Where there is a periplasmic intermediate, we have this first step, the translocation across the cytoplasmic membrane, and a variety of systems, which we'll go into a couple of these in more detail to do that. And then there's a second step where they get across the outer membrane. And then there are various systems for doing that. Type 2 secretion, type 5 secretion, the chaperone-Usher system. These all have methods for translocating across the outer membrane. So starting with the most fundamental secretion system, the sec-translocon. This is ubiquitous and it's essential. It's found in all the domains of life, the 3 domains of life. It's found in eukaryotic organelles as well. We talk about sec-dependent secretion for secretion that is dependent on this particular system. So obviously in gram-positives that is just secretion. In gram-negatives it's translocation across the cytoplasmic membrane we're talking about. Now the proteins that go through this system have to be unfolded. So in fact they're often going through the system quite soon after they've been synthesized, and there may even be coupling of protein synthesis to export through the system. These proteins have a characteristic signal peptide, or leader sequence as it's called, which targets them to the system. It is very easy to recognize in terms of kind of bioinformatics signatures. The transport across this system requires hydrolysis of ATP. It's ATP dependent. And as part of the secretion mechanism, the signal peptide gets cleaved off. Now, this sec-dependent system was often shoehorned into something called the general secretory pathway, tied in with type 2 secretion and sometimes type 4 secretion. So you may sometimes see that term used. Now, there are a number of players in this game. If we look at how stuff is getting across that inner membrane, there is this membrane spanning complex, sec-YEG. There is a series of proteins which are involved in targeting these proteins for secretion. And then there's sec-A, which is the ATPase, which actually energizes this process and drives the proteins through the sec-YEG pore. So we often talk about the translocase as this whole complex that's involved in pushing the proteins through. This trimer of sec-YEG forming a channel in the membrane, and then sec-A as the ATPase. And that binds to this complex of sec-YEG. There are other proteins when we do screens for protein-protein interactions and dependencies of the system. There are other proteins involved. Sec-DF, there's YAG-C, YID-C. These are all involved in enhancing translocation. Sometimes their precise roles are not clear. That's not all been sorted out yet, although there has been some progress.